like the hardest part. Well, no, the next part is reuptake is. So, and I have, you know what, I'll post my other, I have like a past lecture from like last year of this that I still have. So I'll post that. Okay, so there's plenty of neurotransmitters that are involved in psychological difficulties and that are involved in, you know, healthy reactions to things. So if people have a deficit of that, we can increase them, right? And so this is where that happens. That's why it's so important. Okay. And so a lot of study has gone into like which receptors are key in making sure dopamine travels through the brain correctly, and making sure serotonin travels through the brain correctly, that sort of thing. Okay, now this part is hard. It's hard for me to explain, never mind for you guys to understand. So this is how an action potential works. So an action potential is the signal that travels down an axon to the next neuron. Okay, so it's the thing that goes down that long axon, and it has to be strong enough to make neurotransmitters jump out into that synapse and get sent to the next neuron. Okay, but what makes that kind of electrical impulse go down an axon, right? So you guys have heard about homeostasis, right? Homeostasis is the tendency for things to like to stay at in a certain state, right? So we like to be at a certain temperature. You know, you probably like your room, wherever you stay most of the time, to be a certain temperature. I have like a half of a degree window of comfort. Like it has to be exactly like 69 degrees to 70, or I'm hot or I'm freezing. Anybody else like that? Um, maybe it's just me. Anyway, so that's homeostasis. And, you know, the example they always give is like a thermostat. So if the temperature goes above what somebody has set it to, you know, the thermostat regulates it down, regulates it so the temperature will go back down to the preferred resting state, right? So an axon has a preferred resting state, and it's a slightly negative charge. Okay. But when a neuron receives a signal at the dendrites from another cell, that means that sodium, which is Na+, this rushes in, okay, and it makes that neuron inside the cell more positive, right? Because it's a positively charged chemical, right? And so that makes it unbalanced, right? And it likes homeostasis. So now it's too positively charged inside the cell. So something has to happen to get it back to its preferred resting potential, right? So can anybody tell me what happens? What happens? Any biology people in here? What happens to that cell? What does it do to kind of deal with being too positively charged inside?
So it's going to balance. So it's going to diffuse to make the balance less more equal.
Right. It's going to try to make the balance more equal. And to do that, it opens its potassium gates. So it has like door. I always think of it as, well, it's gates. I think of it as literal gates, you know. So these gates open on like the membrane of this action potential and let the potassium out. And potassium is positively charged, right? And the potassium is the K+, right? Let me make sure. Yeah. Yeah, K plus is potassium. So when it lets the K plus out, it's letting the positive charge out. And that allows the inside of the cell to go back to a slightly negative charge. But something happens first. At first, it hyperpolarizes. And what does that mean? What does it mean when the cell hyperpolarizes at first? And you guys, you can have the reading open in another window if you want, and like kind of read through and give me these answers. That's not cheating. That's like we're studying together. This stuff's
It
hard
becomes
to memorize.
it becomes more negative.
Yeah. So when it hyperpolarizes, it becomes more negative than it likes to be for a while. So do you ever go like, I'm cold, and then you crank the heat, and then you forget and you're sweating? Like an hour later, you've hyperpolarized your room, right? So you gotta, so it's like this, and then you gotta let it balance out, right? So that happens to your cells, right? And then it finally goes back to its slightly more negative resting potential. Oh, and here's a little, so it likes to be here. It's like negative 67 or something of a charge when there's like the right kind of, there's all different kinds of chemicals in your cells. So there's the right amount of sodium, potassium in your neurons when it's all perfect and nothing's happening. Well, it's not really perfect because then there's no communication going on between your brain cells. But when certain cells in your brain are quiet, they're resting, it's negative 67 something is the charge, right around here. See how it says, well, it says negative 70, but it's really a little bit lower than that. And then right when enough sodium comes in from a sending neuron, it starts to say, whoa, something's wrong. It's too negative inside our cell. We have to open some gates and let some of this potassium out. This is uncomfortable, right? So the potassium floods out, and that's the peak of the action potential. And at that point, it's too positive outside the cell and too negative inside the cell. Right? And then it eventually goes back to normal. Oh, wait a minute. Now, I'm not describing this right. At the peak action potential, it's positively charged. Oh, inside the cell, because they've let too much potassium get out. Sorry. And then once a lot of potassium gets out, it's like for a little while, it's just right. And then too much potassium gets out and it's hyperpolarized into negative inside the cell for a little while. Sorry, it's so hard for me to understand and explain this. I'm not a natural biologist. I think I have yeah. Thank God for videos. I have a little video of this. Let me hopefully it will share. Let's see. I have to. Can you see YouTube right now? No, okay, so I gotta stop share and then start share again. Am I recording now? Does it look like it's recording? Okay, there's like no sign of it anywhere here. Oh yeah, okay. Okay, stop share. Share screen. Okay, can you see it now, YouTube? Okay.
Yeah, we can.
Where to go? I can't. All right. Is it there?
The human brain receives millions of gigabytes of information every day, and we perform thousands of actions as reactions to these data. We receive information from the external world via our five senses. For example, when we are driving a car, we experience our surroundings primarily through our eyes, which keeps us on the road without hitting anything. The brain is able to receive this information, integrate it with existing information, and direct specific behavioral output. So, how is this possible? The human brain contains billions of neurons, each connected to several thousand other neurons. These neurons communicate with each other through electrical and chemical signals. Each neuron consists of the same basic structure, the cell body, which contains the nucleus and important genetic information, the dendrites that receive information from other neurons, and an axon, which sends information down to its multiple terminals, or end regions, which are known as the terminal buttons. The part where the cell body connects with the axon is termed the axon hillock. Each neuron holds an electrical charge. When a neuron is in a resting state, the charge is about minus 70 millivolts. This is mainly due to the difference in concentration of ions, essentially positively or negatively charged particles, inside and outside the cell. The outside of the cell has a high concentration of positively charged sodium ions, as well as negatively charged chloride ions, more commonly known as salt or sodium chloride. The inside of the cell has a smaller amount of positively charged potassium ions and a larger quantity of negatively charged protein ions. Embedded in the surface of the cell membrane, which separates the inside of the cell from the outside, are protein structures known as ion channels. These channels act as gates by both permitting and hindering the flow of ions into and out of the cell. Normally, these ion channels are held closed, maintaining the electrical potential of the cell. However, they can be opened if the right message is received from a neighboring neuron. Each neuron can receive inputs from thousands of other neurons, and the place where one neuron makes a connection with another neuron is called the synapse. When a neuron releases a chemical signal known as a neurotransmitter, NT, into the synapse, the chemical will bind to another type of protein embedded in the membrane of the receiving neuron, known as a receptor. Once the neurotransmitter binds to the receptor, much like a key into a lock, this leads to a number of other biochemical events. One possibility is that this receptor binding will open an ion channel. This binding may also lead to a sodium channel opening, allowing the passage of positive sodium ions into the cell. As these ions are positively charged, the area around the channel will also become more positively charged. This small change in the membrane is called an excitatory postsynaptic potential, EPSPs. Similarly, it is also possible that part of the cell becomes more negative, for example, by influx of negatively charged ions. This is called an inhibitory postsynaptic potential. If enough positively charged ions enter the cell, thereby resulting in more excitatory postsynaptic potentials, and the membrane potential reaches a threshold of excitation at the axon hillock, this will trigger an action potential. The action potential is essentially a brief increase in the permeability of the membrane to sodium, immediately followed by a brief increase in the permeability of the membrane to potassium. Once an action potential is generated, it propagates all the way down the axon to the terminal buttons. In this sense, action potentials fire in an all or nothing fashion. Once triggered, it will continue all the way down the length of the axon to the terminal buttons. It is important to note that while EPSPs are small, local, and contain graded changes in the membrane potential from more negative to more positive, an action potential is a brief but larger reversal in the membrane polarity. Once the action potential reaches the terminal buttons, this change in membrane polarity causes calcium channels, normally held closed, to open and allow calcium into the cell. Calcium then acts as a signal to release the neurotransmitter substance, which is docked at the presynaptic membrane via exocytosis. The neurotransmitter then binds to the postsynaptic receptor, and then the cycle begins again. So the cells in our eyes receive input and send a message to the cells in our motor cortex to send yet another signal to the cells connected to our leg muscles that will hit the brakes.
So they give the example of, motor coordination and vision, but, the same kind of synapses occur to create an emotional response, to stimulate like a memory when you smell a certain thing, for like a memory to rush back, for a dream to occur. So lots of things that go on psychologically. Not those things are considered psychological things too. Vision, coordination, motor skills, those are all really important parts of psychology. So if you're interested in studying those things, you could become a psychologist and spend your time studying those things. But there are other things that you more classically think of as psychology that involve action potentials, which are a vital part of communication between brain cells and between any neurons. Okay, so how does this affect how we use medications in, here's my, how we use medications in psychology? Well, in a couple different ways. So just find one way is we can change reuptake between two cells. So can anybody tell me a type of medication that reuptake is definitely involved in? Oh, I seem to have forwarded through the entire. So, Kate?
SSRIs.
Yes, SSRIs, which are selective serotonin reuptake inhibitors, meaning they stop or inhibit reuptake, not all of it, but they inhibit it. So they slow it down. And can someone tell me what serotonin plays a role in? Like what kind of things? What's serotonin implicated in?
It stabilizes our mood.
Yeah. Serotonin has a lot to do with our mood. And remember, I was talking about ruminating. Serotonin helps us regulate ourselves so we do less ruminating. So there are a lot of disorders that it's been found to be very effective for. So things like depression, anxiety, obsessive compulsive disorder, premenstrual dysphoric disorder. So disorders where a person will focus on things that are a source of worry or a source of negative feelings, a source of humiliation or shame, whatever, and it's really hard to stop. And serotonin seems to give people like more of a choice. There's still a big part of it that's up to the person, but it's less. The person feels more in control when they have more serotonin in their brain. Okay, now it's not going to make somebody who doesn't have a serotonin deficit feel more powerful or something. But if somebody has one of these disorders, they're going to feel healthier if they take a medication that moderates the amount of serotonin in the brain. So what reuptake is after the sending neuron sends some serotonin, we'll say, to the to the next neuron, some of it stays in this area here called the synapse, the space between two neurons. And then after a while, some of it comes back up into the sending neuron. So it's taking it back. It's reuptake. It's going back up into the neuron that sent it in the first place. So think, you go to a party, you hand out serotonin cupcakes. Not everybody takes one. You leave a few there for the next class, say, and then you take the rest home, right? That's serotonin reuptake cupcake. Cupcake reuptake. All right. But yeah, that's what reuptake is. And you can inhibit that with medication, okay, with SSRIs. And they don't know exactly why that makes people feel better. Some people think that's because it tricks the sending neurons into creating and sending out more serotonin. Because think about it, if you're getting a lot back, if you're not getting a lot back because this person has a diminished serotonin productivity because it's inhibiting the reuptake, the cell thinks it needs to produce more. Okay, so let me explain this better. There's a medication that stops this reuptake from happening to the same extent, right? So say you go to your party and you have your serotonin cupcakes, but this time and last time you had like 10 left, right? And then you go to another party and you only had one left. You're like, whoa, I better make a few extra serotonin cupcakes this time, because what's more humiliating than not making enough, right? So if you're if you think of this sending neuron as the cupcake maker, it's getting tricked into making more serotonin. Does that make sense to you? Because if there's not any coming back, it's thinking, not that, they don't have like existential angst or anything, but it's tricked into thinking, I need to send more out because nothing's coming back. How weird, right? And then some people think that, because it's blocking these little doors that let it take the serotonin back in, that there's just more serotonin in these synapses than there would be without it. And that's why people feel better. It could be both. It could be making more serotonin overall in the brain. And it could be also that it's making it so more of it is hanging out in these areas in between the neurons. Do people get that? So serotonin's, good. You can get serotonin syndrome, which is not too good if you take certain medications and you have too much serotonin. That's not good, but it's super rare. So in general, serotonin tends to help people with depression, anxiety, obsessional symptoms.
Professor, I have a question.
Uh-huh.
So what happens if there's no reuptake or there's not like binding into a receptors for I'm sorry, but they say like in between the space in the postsynaptic space, like what happens there?
If they stay in the synapse?
Yeah.
Well, it's like they think that if there's enough sending neurons sending serotonin out into the synapse, then people will feel okay. That it doesn't have, there is a serotonin pathway that goes around the limbic system. And I get, in order for it to be sent out, there has to be an action potential from a sending neuron. So there's got to be at least some connections for it to happen at all, right? Even if there's no reuptake, for it to have made these vesicles burst at all, an action potential must have happened, right? Which means that there was a signal coming in through the dendrites, right? of the sending cell in the first place. So there's some kind of connection going on. But even if it's not like thousands of connections, those neurotransmitters in the synapses make people feel good. Serotonin makes people feel at least functional. People don't get high from serotonin. What can people feel high from? What neurotransmitter can people feel high from? Can somebody think of one? Kate?
Dopamine.
Yeah, yeah, hence the term dope, right? The term dope comes from dopamine, right? We didn't name dopamine after dope. We named dope after dopamine, right? And can somebody tell me what dopamine is implicated in? What psychological disorder? There's a dopamine hypothesis for this disorder. It's a very severe one.
Schizophrenia. Like ADHD.
I'm sorry, I didn't understand.
ADHD. Oh, sorry.
PTSD, no. I mean, it probably plays a role, but there's one that has a clear hypothesis just based on this.
Is it in is it for schizophrenia?
Yeah, schizophrenia. Yeah, and there's a bunch of reasons why they first came up with this. They used to give jet flyers, why can't I talk, who would do bombing missions in World War II, who would have to like fly for a really long time to get to Europe, bomb Europe, parts of Europe, and come back. They used to give them speed, which increases the amount of dopamine in the brain, and eventually, to keep them awake. I forgot to mention that very key point. And then, but eventually they'd start exhibiting signs of strange behavior that looked like schizophrenia. So they thought, maybe it's, the excessive amounts of dopamine in the brain that causes schizophrenia. Right? Another thing was, has anybody seen Awakenings with Robin Williams? Yeah, so that's based on the work of Oliver Sacks, a famous neuropsychiatrist, who was working with people who had syphilis, who had a kind of psychiatric illness at the end of their lives where they had like a locked in syndrome and couldn't move at all, but there was still somebody in there, mentally. And he would give them a synthetic form of dopamine and they would wake up and be like their old selves. But then over time, after enough dopamine built up in their system, they started acting really unstable and like somebody who had schizophrenia. So these are the places that the schizophrenia that the dopamine hypothesis of schizophrenia came from. Right? So that's reuptake. So there are different ways that drugs can impact neurotransmitter imbalances. Agonists mimic a neurotransmitter, so if you take something that mimics a neurotransmitter, you're going to feel like you would if that neurotransmitter was in your brain, right? So some examples of agonists are opioids. So, because opioid drugs activate the opioid receptors in the brain, and they result in a full opioid effect. That's why they're so addictive. So hydrocodone, oxycodone, heroin, morphine, opium, methadone. That's why those are so addictive, because they act directly on the opioid receptors, and people feel exactly like they do when their body's own pain receptors would be activated. but it's immediately quick. It's extremely quick and it's extremely strong in effect, right? So agonists make you feel like you have more of that neurotransmitter in your body. And then antagonists block the normal activity of a neurotransmitter or a receptor. So they might kind of look like the key, but just go into that receptor and make it so nothing else can go in. So there's no connections happening over the synapses, right? There's no connection between any of the neurons because there's these things that kind of look like keys, but they're actually blocking the keyholes, right? They're kind of like child safety locks, right? So you can't, like a big thing over the doorknob so you can't get in, right? That's what antagonists do. So agonists, increase the activity of receptors for a neurotransmitter, and antagonists block it. When somebody's antagonizing you, they're making it so you can't do your work. You can't go about your daily activity, right? And that's what antagonist drugs do. They make it so that these certain neurotransmitters can't work. So dopamine can't have its usual effects, which would be a good thing in schizophrenia to slow down hallucinations, right? All right, so I think that's the last one before the nervous system. Yeah, so I think I'm gonna stop there. So I hope you guys have a good day. Any questions or anything do you guys want me to keep sending you links to the recordings? Are those helpful? Or is anybody actually looking at them? Like, do they really look at class again after they come to class? Okay, so I'll keep doing that. I think I'm a couple behind, but I will do that. All right, take care. Have a good day.
Thank you.
Thanks.
You.
Thanks.